Academy, and this is Dr. Anita Srivastav. Today I'm going to talk about downstream processing. So before starting with the downstream processing, I will tell about the total process that is involved in the formation of an industrial product, which involves the role of the microorganisms, or basically if I talk about in industrial microbiology. So if I talk about downstream processing, so basically downstream processing, it has two processes which are upstream processing as well as downstream processing. So before going into the detail of the downstream processing, I will brief about the downstream process. So downstream process, it refers to the first step in which the biomolecules, they are grown in bioreactors, and once they reach the desired density, that is through the batch and fed batch cultures, they are harvested and moved to the downstream sections of the bioprocess, and it includes all steps related to the development of microorganisms, that is nutrient preparation, cell culture, cell separation and harvesting. So the two stages of industrial fermentations which I said are upstream and downstream processing. So upstream involves all factors and processes of the fermentation. Then the different stages of processing which occurs after the completion of the fermentation or bioconversions. These are the separation, purification and packaging of the product. Means at this stage I am talking about the downstream processing which ends or which starts after the ending of the upstream processes, and the different processes which are used for recovery and purification of biosynthetic products, particularly if I talk about the food, the biopharmaceuticals, biofuels, and these all are defined under the downstream processing. The process also includes the proper treatment and disposal of waste as well as recycling of the components. It means during the formation of any product or during any industrial processes, as the process is done at the industrial scale, so a large amount of disposable or the waste products are also generated. So it is very important to to dispose that waste material in a safe manner. So therefore the strategies includes the disposable of these waste, as well as it is also important to recycle these components. So it is also an important factor of the industrial process for the disposable of the waste as well as its recycling. Now talking about this is just what I said about the upstream process, that it includes fermentation medium formulation, fermentation medium, inoculum preparation, medium inoculation and air sterilization. So the fermentation medium which contains an energy source, that is a carbon source, a nitrogen source and micronutrients which are required for the growth of the microorganisms. So a medium that is used for a large scale fermentation, it should have the following characteristics. So first one is economical and easily available. That means the fermentation medium which is used during the process, or the carbon sources, the substrate which we are using in this medium, they should be easily available and should be economical. That means they should not add any extra cost to the industrial process. Then enhancement of growth of the microorganism, productivity and the rate of formation of the desired product. So these three things, basically talking about the growth of the microorganisms, about the productivity and rate of the formation of desired product, It means the process, it should be designed in such a way, the upstream process, it should be designed in such a way that the microorganisms which we are going to use in the process, uh, basically, which is going to be used in the process, it should easily utilize that medium so that the growth may be enhanced and the cell density may increase. Therefore, the second thing, the productivity. So, if the enhancement of the growth of the microorganisms takes place, the productivity will also be increased, and therefore the formation of the desired product will be high, or the rate of its formation will be high. Then reduce the formation of undesired products. So as we all know, as I discussed in other presentation also, that the basically 70% of any uh, product or the industrial product cost is dedicated to the downstream processes. Downstream processes means we are talking about the purification and recovery as well as packaging. So if the undesired products will be more, means more steps will be done for the removal of these undesired desired products, and in turn it will add to the cost of the process. So the formation of the undesired product should be minimized during the process. Now talking about the main content, that is the downstream process. So as I told that once the fermentation run is finished and once the cell or the product is being formed, the fermentation broth, it is processed for the recovery of the desired product. So basically the main aim of the downstream processing is to provide an efficient, reproducible, and safe recovery of the target product to the required specifications, that is biological activity and purity, etc. It means the downstream process should be efficient, reproducible, and safe. And the most important feature is that during the recovery of the target product, the biological activity as well as the purity should not be compromised, means there should be minimum loss of the compound, and the biological activity should not be disturbed, should not be disturbed. Then maximizing the recovery yield and minimizing cost, that means the process should have the potential to maximize the recovery yield, means the recovery should be maximized, it should be in a maximum form. And minimizing cost means the process should involve minimum cost. So the target product, it will be recovered by processing of the cells or spent medium, depending upon whether it is an intracellular or extracellular product. So now the product may be intracellular or it may be extracellular. So the steps involved in this process will totally depend upon the kind of the product we have. If we have the intracellular product, then after the cell harvesting, the cells will be processed for its, like they will be disrupted. And if it is an extracellular product, the basically the medium of interest will be the fermented broth. Then the end products which are formed, they may include the antibiotics, amino acid, vitamins, organic acid, industrial enzymes, vaccine. So talking about the industrial products in industrial biology involving the microorganisms, these are the main products on which we are going to basic means I mean to say that industrial microbiology depends on the formation of these products during the fermentation. then it is complex and important as fermentation process. So this downstream process is equally important and complex as the fermentation process. Now talking about the factors involved or the strategies that are involved in upstream process. So upstream processes we are going to talk about, as I discussed earlier also, it is the involvement of the microorganisms and the fermentation raw material. So if we talk about the microorganisms, so first of all initial isolation, then strain improvement, means the isolated strain should be improved, or basically if we have any production strain, the strain improvement can be done with it. And about I have talked in my earlier video about the industrial microorganism strain improvement, but the basic constraints uh, include the nutritional requirements, metabolic controls, shear sensitivity, temperature optima, morphology, oxygen carbon dioxide effects and requirements, genetic stability, metabolic byproducts, viscosity effects. So uh, after this process, starter culture propagations, it here I will talk about this only, but here I will pick with the fermentation raw materials, which consists of the sources of the carbon, nitrogen, phosphorus and sulfur. It also includes some minor elements like trace elements, growth factors, water, etc. And the availability, cost, stability and pre-treatment and sterilization requirements are also very important factors that are to be considered during any upstream processes. So including all these things, the media development takes place, and then it can be talked in two forms, that is the propagation medium or the maintenance medium. And here we have the production medium. So talking about the propagation medium, we are talking about the starter culture propagation basically. So it can also enter here. Then if we talk about the production medium where we discussed about all these components. So medium development and then the production medium is taking place. So now during a fermentation process, basically this is a bioreactor where the fermentation is taking place. So this fermentation is taking place in under the controlled environment, under the control factors, that is the oxygen, pH control, anti-foam, and with the involvement of the cooling or the heating systems. So this is fermentation, and here basically, and here it is also taken care of the supported or the suspended growth, fermenter type, the kind of fermenter which we are going to use, the stirring mechanisms, site geometry, mode of operation, instrumentation and automation. So these things we are talking about this in the form of the bioreactor, that the kind of the bioreactor which we are going to use. So these are very important factors which we are talking about. Now, after the upstream process, I'll be talking about the downstream processing. So, in the downstream processing, here we can see that the downstream process, it is influenced by product concentration and stability, and the other considerations are yield at each step, the process cost and purity requirements. So, after the upstream process, the downstream process can be in situ or ex situ. If I talk about the in situ DSP, that means I'm talking about some integrated systems that are assembled within the fermenter, where the production as well as the separation can take place. So this is actually the in situ DSP. Where if I talk about the XC to DSP, that means after the run of the fermenter, the media is taken out and it is processed outside the bioreactor. So first step is cell separation. Cell separation may be done by centrifugation or filtration. So here we can see that the harvested cells, we so after, see in the bioreactor, the processed broth we have, so it contains cell as well as the harvested cells as well as the spent medium. So if we are taking the harvested cells and here we are taking the spent medium, so harvested cell means if the product is extracellular, that means we do not require the cell. But if the product is intracellular, then the harvested cell, intercellular or plasmic product is there, then the cell disruption will take place, and the method of the cell disruption of the cell breakage I will discuss in the coming slides about this process, about this step. Then after the cell disruption, centrifugation or the ultrafiltration will take place, by which we will get the cell-free extract and the inclusion bodies, and cell debris will be discarded off. But if the product is extracellular, if it is spent medium is of interest, if the product is extracellular. So after getting this extracellular product, we are going to concentrate this uh, this broth or the fermented medium, and there are about the concentration step also I will discuss it on later in the coming slides. Then this medium concentrate, it is being processed for the purification by dialysis, precipitation, partition, chromatographic steps, ultrafiltration, and even distillation, extract, etc. Then after the purification, the process of crystallization, drying, lyophilization, sterile filtration, and packaging. So this is basically the getting it is a part of the packaging. Means after the concentration, purification, then it is to be packaged, and then finally we are going to have a finished product. So as I told that the downstream process, I will just brief it. Downstream process, it is divided into three parts. One is the primary recovery, then the product purification, then the finishing processes. So this is the brief explanation about the downstream process. Now I'm going to discuss the steps involved in the downstream processing in detail. So first step is the cell separation, second step is cell disintegration, then we have extraction, concentration, purification, and finally drying and packaging. And this is in short also the basic operations, that is first is fermentation broth, raw product, final product. So when we talk about the fermentation broth, it is the removal of the biomass and other solids, as I discussed earlier. And this includes the method like centrifugation, filtration, sedimentation, or sieving. And talking about the concentration and fractionation, so it involves extraction, ion exchange, adsorption, precipitation, as well as distillation. Final product, the basic operation is final purification. After the final purification, we are going to have the final product, and this final purification includes chromatography, distillation, decolorization, crystallization, and drying. So these are the basic steps in very short, just to understand about the downstream process. Now separation of the particles. So basically there are three methods to separate the cells from the processed fermentation medium. First is the flocculation and flotation. The another is other is the centrifugation and filtration. So flocculation, basically it is the process by which individual particles aggregate to form a clump by adding a clarifying agent. And flotation is the process of separation of particles based on relative buoyancy of particles. Froth flotation, dissolved air flotation, or induced gas flotations. Then moving to centrifugation, it may be used to separate bacteria and usually protein precipitates. And the centrifugation, it works on the sedimentation principle, involving the centripetal acceleration, which causes denser substances and particles to move outward, and less dense are displaced and move to the center. But difficulty arises due to small differences in the density of the particles and the medium. In that way, there is some difficulty during the centrifugation. Then the third step is filtration. In filtration, it is used actually for the separation of the microbial cells. Different techniques used for filtration are centrifugal filtration, cross flow filtration, microfiltration, ultrafiltration, surface filtration, rotary drum vacuum filtration. These are actually the different techniques which we are going to use. So here I am not going to discuss this in detail. It is just about the information that these processes can be done during the separation of the particles, and that these may be discussed later on. Now talking about the cell disruption. Cell disruption. So does cell disruption is it is a process by which intracellular fluid is obtained by breaking the cell wall. Now the purpose for the cell disruption is to obtain the intracellular fluid without affecting its components. So now it is very important. It is very important to understand that the process is to be done where the intracellular components are not affected. If the intracellular components are affected, that means there will be some kind of loss, or some kind of means the final product is going to be affected if this process is disturbed. Then the recovered product should not be affected. This is also important, that the product or the desired product or the targeted product, so the recovered product should not be affected. Now there are two types of cell disruption methods. One is mechanical and the other is non-mechanical method. Mechanical methods, it is divided into solid shear methods and liquid shear methods. Whereas non-mechanical methods, it can be divided into physical methods, chemical methods, as well as enzymatic methods. So these are the two types of cell disruption, mechanical and non-mechanical. Now I'm going to tell in detail. So mechanical methods, it involves shear forces in solid matter and solution. Then non-mechanical methods includes lysis. Now the the kind of lysis that can be done are physical. In physical, we are going to talk about the freezing or thawing. Now high osmotic pressure and shock. Then when we talk about the chemical, it is surface active agents, solvent, antibiotics, etc. Enzymatic, example, lysozymes. And talking about the non-mechanical methods, we have drying also. So in drying, it is freeze drying, air drying, pressure release drying with solvents. Now talking about the extraction. So after the harvesting of the cells, the extraction step is followed. Now liquid extraction. Here I am going to talk about the liquid extraction. So this liquid extraction, it is a separation process which takes the advantage of the relative solubilities of solute in immiscible solvents, and the solute is dissolved. It is dissolved more readily, and it becomes more concentrated in the solvent in which it has a higher solubility. Then a partial separation occurs when a number of solutes which have a different relative solubilities in the two solvents used. Solvent should be non-toxic. It is very important that the solvent should be not toxic, selective, inexpensive, and immiscible with broth, and it should have a high distribution coefficient for the product. So these are some characteristic features about the liquid extraction method. Now after the extraction comes the step of the concentrations. So now there are certain methods, there are some types of concentration methods, which includes evaporation, membrane filtration, chromatography techniques, and here especially I am talking about the ion exchange chromatography. Then we have the adsorption. So these are four methods which are used for the concentration of the product. Now purification of the product. So for the purification of the product, there are different chromatography techniques which are used. Like we have the paper chromatography, thin layer chromatography, column chromatography, ion exchange chromatography, affinity chromatography, high performance liquid chromatography, reverse phase high performance liquid chromatography, then gas chromatography or the mass spectrometry, gel filtration chromatography, or we can say size exclusion chromatography. So now, as we all know that chromatography is basically, it is based on the principle of partition of solute between two phases, and it usually consists of a mobile phase and a stationary phase. So the mobile phase, it usually refers to the mixture of substances to be separated, dissolved in a liquid or gas. And the stationary phase, it is a porous solid material through which the sample contained in the mobile phase percolates. So in paper chromatography, a paper is used which has a stationary phase. It contains cellulose, and a sample which is to be detected. It is placed on the paper, and then it is we are using the mobile phase. Uh, then it moves with the help of the mobile phase the compound which are to be separated. In thin layer chromatography, we are using the solid support like silica gel, and which acts as a stationary phase, and the compound of interest, it is spotted on this thin layer chromatography. And then we are using a solvent which is a mobile phase, and then the material can be detected, and by these process by these chromatography, we can detect the amino acids, some sugars. Now talking about the column chromatography, so the most importantly used column chromatography is basically the high performance, this HPLC we are using. Then we have ion exchange chromatography, and gas chromatography also. So talking about the liquid chromatography, here the separation by liquid chromatography is based on the distribution of the solutes between a liquid and mobile phase and a stationary phase. And when particles of small diameter are used as stationary phase support the techniques, it is HPLC. The most widely used liquid chromatography is HPLC. So here, if I talk about the some instrumentation of this HPLC, actually a basically a solvent reservoir is used to hold the mobile phase through the system, and there's an injector which is used to introduce the sample into the column, and we have a chromatographic column which separates the solute, and we have a detector to detect the separated analytes as they are eluted from the column, and then a computer is attached which processes the system and processes data. So there are there are this is few examples of the chromatography which we are going to use, and one more important is ion exchange chromatography also. And this ion exchange chromatography, it is based on an exchange of ions between a charged stationary surface and ions of the opposite charge in mobile phase. And then depending on the conditions, solutes are either cations, that is positively charged, or anions, that is negatively charged, and these are known as the ion exchangers. So now after the purification product, means this purification only, basically the purpose of this product purification, it is done to separate those contaminants which resemble the product in physical and chemical properties, and they are very sensitive and sophisticated equipments which are used. The process is expensive to be carried out. Then the product is purified by using chromatography techniques. Now the final product processing. So the final product processing includes crystallization, product stabilization, and drying. So talking about the crystallization, it is a process of formation of solid crystals by precipitating from a solution, melt, or more rarely deposited directly from a gas. So this is the process of crystallization. And chemical solid liquid separation technique, in which mass transfer of a solute from the liquid solution takes place to a pure solid crystalline phase occurs. Then product stabilization to avoid premature degradation or denaturation. So this product stabilization is required to avoid this premature degradation or the denaturation. Then drying involves the removal of water from a solid product. And finally, filling, labeling, and packaging takes place to prepare product for use. So these are the final product processing involving crystallization, product stabilization, and drying. Now this is the basic outline of the general steps that is taking place in downstream purification. So from the fermentation breath, from fermentation broth, where the cells are there or cells are not required. So here we can see the cell separation, cell disruption, debris separation, precipitation, and if it is intracellular, so intracellular, these steps these steps are involving. In case of extracellular, directly the concentration is taking place, and then high resolution purification, then finally the polishing of the product. So this is the purification step, and there here finally we can have the recovered product. So thank you.